No. Can you? Yeah. Can you see the presentation screen? Yes.
Yes.
Good. Thanks. Well, so thanks for the introduction. Today we'll show you what we are doing, especially for organoids and 3D cell culture. I'm part of the functional neuroconnectomics group with Martin Schwartz in the University Hospital in Bonn. And well, we are microscopy team. So as you know, right, depending what you would like to see, right, in terms of size, you will choose different methods. We use light microscopy, but unfortunately we have this diffraction limit of the light that is about 250 nanometers. That's why there are like some developments in super resolution microscopy, like STED, as was mentioned before. And we are interested in neuronal interactions, right? And neuronal interactions are usually in the range of 100 nanometers, where the like synaptic distances occur. But still, we would like to see the complete context of these synaptic connections. And if we have like a mouse brain or a big organoid samples, we can have samples in the range of 1 centimeter. So what we can do in order to get this detailed image with subcellular features. So first, let's have a look like what's happening under the microscope, right? So we have a point source. After the objectives and all the lenses, that point source doesn't look like a point source anymore. So it is more blurry, it's distorted. So that's why when we have two elements that are too close to each other, the PSF, right, this distortion, it's overlapping and it's difficult to differentiate. So we have these super resolution methods that try to sharpen or play with some statistics in order to differentiate these two elements that are too close to each other. But another method, right, and that is cheaper, is expansion microscopy, where we physically separate the elements in order to resolve them. So how expansion microscopy works, I mean, was also explained last week on Friday in several talks, right? So we have one sample here, we have a cell, and we would like to have that cell bigger. And let's say that we want to see the microtubules, then we have to label that either with antibodies or we genetically encode like a fluorescent protein. Then we have the linker that is in one side catching this basic amine groups, and in the other one will get attached to the polyacrylamide network that we are inserting into the sample. And then the digestion or homogenization part, where we take out everything that is not linked to the gel. And because this gel is basically a composition between acrylamide and sodium acrylate, right, have this negative charge that when you put the gel in water will absorb water molecules that acts as a dipole and will get attracted by the negative charge of the gel, and we will have this physical expansion of the sample. So in the lower part, you can see an example, right, of mouse brain slice, how that looks after expansion. The advantage of expansion is that we will get a nice transparent sample, and we have refractive index like water. So that's probably one of the main differences also with other clearing methods. But the original protocol was usually limited to a thickness of 100 microns, more or less. And the preservation of fluorescent proteins was not properly done. I mean, then later was other protocols that allows to do that. We also play basically modifying, for example, the concentration of the linker by 2 times, and also not using just water for the expansion, but we use water together with a little bit of a buffer that is herpes, and always checking that the pH of that was about 7. That allows us to preserve most of the fluorescent proteins with really good signal. The only drawback of that is that the final expansion ratio was reduced from 4.5 to 4, but still you will get an improvement in the resolution of 4 times. Now, how to get the protocol working on thicker samples, well, for that, we optimize the permeabilization of the sample, penetration, incubation steps, and how we do the digestion. So with that, right, we can work with samples that are more than 3 millimeter thick. So that will be 12 millimeter in thickness after expansion, still preserving fluorescent proteins and all the structure. But how to image these big samples, right? Usually you will bring that to a confocal microscope, right? So in confocal, we excite through the lens and we acquire the fluorescence with the same lens. And if we want to scan a plane, then we have to go point by point. That is, I mean, confocal works really good. It's like more or less like a standard in microscopy for biological samples, but it's too slow, especially if we have bigger samples. And also we have the problem that we could have some bleaching. So that's why what we use is light sheet microscopy, where we can decouple the illumination and the acquisition lens, right? Light sheet microscopy is an old technique, is from the 1900, but was just becoming popular in the after 2004. And what we do, right, is that we have the one illumination objective and one acquisition. And like that, we generate a plane, right? Just where we want to take the optical section, and we can acquire that at once, right? So that in that sense is faster than confocal. Other advantage of light sheet that we can see here, right? And these are epifluorescence example where we have dextran molecules attached with a fluorophore that is Ato 633. And with epifluorescence, even if the acquisition is also quite fast because we acquire the complete plane, the contrast is not really good. On the other side, we can see what is happening with light sheet. So where we have like just one plane of illumination, we can clearly see the molecules. We can even see these airy patterns when they move out of the focal plane. And so when we compare these two type of illumination, we can see that we have like a higher contrast, right? So in general, light sheet is really fast, will bleach really little your sample, but have one drawback that is really sensitive to the absorption of the light. So that means that if your sample is not transparent, then probably light sheet is not the way to go. Luckily, with expansion, we have a transparent sample, and we will have a big sample. That's why the combination with light sheet and the characteristics is something that was published by the group some years ago. And so the combination of these two techniques, right, it goes like really, really well, especially for big samples. The only problem is that because we have a bigger sample, we will also be generating, we will generate big data if we want to take everything with the highest resolution. So in this table, we can see at the very end, right, that if we want to acquire 1 cubic millimeter with the best resolution that we have in that light sheet, it's a custom built setup where we have an effective resolution of 90 nanometer after expansion. You will be generating about 8 terabytes of data only in one channel. So, and that is, of course, not ideal if we want to see a big sample. So that's why we recommend to have like a multi-scale approach, playing a little bit with the expansion factors and the objectives of your microscope. And that's something that I will show you with some examples now. So let's say that we want to have the complete information of the organoid in 3D, but also be able to have cellular and subcellular resolution. So we need to be able to preserve the 3D architecture, be able to image the complete organoid, and still have super resolution features. So this is more or less how the pipeline looks like in our case. So we recommend the use of chaps for the permeabilization. I will show you later why. But until that, and the staining or labeling of your sample is everything normal, like fixed samples with 4% PFA. And then the idea is that to expand your sample 4 times, but if you play with the buffer, you can also have like a 1.5 expansion, for example, if you keep your sample in PBS. Right? So in the upper part, you can see one of organoid that is a bit more than 1 millimeter embedded in the polyacrylamide gel, how the same gel looks after 1.5 expansion 4 times, and an optical section of the sample that we have inside. So if we have this, if we are able to play with the expansion of the sample plus, right, the imaging that with different objectives, then we can address different scales, right? So have a specific resolution for specific scales and get the necessary information. So here we like to show you why CHAPS is a good improvement from Triton. So this is the same type of sample, like cortical human brain organoids that were incubated with the same amount of Triton and CHAPS. And you can see how with CHAPS the penetration of the antibody is better, but you not only get that benefit in terms of like the labeling, but also later when you treat the sample, right? The digestion is usually faster when you use also a good permeabilization beforehand, right? It's like 40% faster than with the normal protocol. Okay. The advantages, of course, of using like light sheet and expansion, we can see here compared to confocal. Even if you have like a nice staining, right, of the complete sample, if you just put the complete like your sample in confocal, at some points you have like absorption of the signal. You are not able to reach the middle part, but when you have a nice and transparent sample, you can reach that. You can get more information. But even if you have then the sample transparent and you put it in the confocal, that's what we have in the upper right part. The working distance of the confocal is limited compared to a light sheet microscope, right? So still we can get more information from the light sheet when we have the sample. Another point that is important, right, is to preserve the like the 3D structure of the sample. So this organoid that was also presenting some GFP neurons. Here we have the GFP signal, so we can see that before the expansion or the clearing part, right, we have a lot of background. After that, the signal is quite clear, and on the right side we can see the comparison of both to see that the distortion is minimal, right? So, and that is really important if we want to analyze what is happening in 3D, that we are not deforming, right, our sample. So we want to see these organoids, right, in the mesoscale in order to see general architecture and cell distribution, right? And here we have like a 2-month-old organoid that was with nuclear staining and Z01 to see the lumen of the rosette area, where is all the cell proliferation and differentiation happening, and we are able to scan this with a low magnification objective, but that volume was still made with several tiles. And we can use, for example, the nuclear staining to get the overall volume or surface of the organoid. And due to the nice penetration, right, because of the permeabilization and the improved protocol, we can then easily do a segmentation of all these lumen from the different rosettes, right? So if we can do a segmentation, means that we can quantify, for example, apical surface area. But that is just one example. I mean, here I can show more parameters that we can already get from the mesoscale using the expansion as a clearing method, right? So we don't have to expand, and we can still get all this information that, for example, allow us to compare organoids under drug treatment or disease models, and so on. Now, if we want to go to the microscale, right, we would like to see subcellular features, and the same sample that was also stained with SOX2, this stem cell marker, we use that to analyze cleavage planes, right? So as we can see here, we have like this horizontal cleavage plane, right, like cell division compared to the lumen area. But because these samples are 3D samples, if you have the same sample and you have a look to the exact cell division from a different angle, you can get a completely different interpretation of that cleavage plane. Right, so that's why it's also important for us to preserve the complete 3D information in order to then focus now in a small region of interest and with a better resolution, right? Is either having a better objective or now having the same, let's say 4x objective, but expanding 4 times the sample, we are able to get information like this, right? Like 3D, the complete 3D information. Of course, we did the validation of this cleavage plane for the corresponding organoid. And what is important is that also here, right, we use the SOX2 antibody to see the cell division and cleavage plane, because due to the resolution, you have to also keep in mind that you will get more information, and that will save you some, for example, with SOX2 and the nuclear staining, we are able to identify the chromosomes and the cell division without the need of a specific marker, like for example, phosphobimetry, right? So one of the advantages also of the using this light chain expansion is that you will get more information out of your sample. Now, if we want to go to the nanoscale, right, if you are interested in super resolution, right, we would like to see the pre and postsynaptic proteins. So for that, we use like now a bigger sample that was organoid incubated for 5 months. You can see that the sample is quite big, so it's like more than 1 square centimeter, 5 millimeter in thickness. So here we will not get the complete image in high resolution, but we will focus only on a region of interest. So here we scan this cubic millimeter, right? That is already, as I mentioned, like about 8 terabytes of data. And here we will focus right in one of the projections where we see some protrusions, right? So from the 2D, you are not, or we're not able to say if those are spines, but because we can scan this nicely in 3D, and we have like this high contrast due to the light sheet microscope, we can then clearly do see, and after a segmentation, right, the dendritic spines that we have in the sample. Unfortunately, after 5 months, this sample was not presenting any pre and postsynaptic signal. So we incubate another one for 14 months. We did the antibody against Homer1 and synapsin. And we focus again in a small region of interest, the one that you can see in the video. We have many of these co-localizations of pre and postsynaptic proteins, but in order to quantify, we choose the GFP that is marked in blue, and then use that as a mask in order to see the pre and postsynaptic proteins that were closer or contained into the sample. Here we have like a multisynaptic button, and we can quantify. So we can quantify spine distribution or synaptic distance. We have like synaptic distances in the order of 150 nanometers, something that we also check with STEM microscopy to validate our method, and was in the same range. The advantage here is that we never cut the sample, right? We just choose a specific region of interest, and if we want, we can then move to another part of the sample and still go with the highest resolution and get more information, right? So we are not cutting also the projections. We are able to follow, right, and reconstruct a complete neuron with the same sample, right? I would like to show you now like a different example that is with assembloids, right? This time it's a project, a collaboration that we did with the university in Luxembourg, where they create these assembloids with a striatum organoid and a midbrain organoid. And in order to keep these two organoids together and generate this assembloid to recreate the nigrostriatal pathway, they have to embed the sample in something that is called Geltrex. And Geltrex is a basement membrane extract, primarily made of laminin, collagen type 4, entactin, and other things. So we have to improve a little bit our digestion steps in order to have this sample transparent. But the idea here is to show you that the protocols, right, or what we have is quite flexible, so you can modify and adjust different digestion steps in order to, again, preserve the 3D information of your sample, right, as we show here. So we have the small green circle that you see there is the midbrain organoid that contains the GFP positive cells, and the rest is the striatum organoid. The Geltrex was there and was visible a little bit in the far red. Still, like if we enhance a lot the brightness and contrast, but it's totally digested and it's not interfering in the sample when we want to acquire this in 3D. So we still get the complete information, right, of this assembloid, and focus now in the interaction between these two. The idea here will be to see if these dopaminergic neurons that are marked in red, so we use a tyrosine hydroxylase antibody, if these ones are sending projections, right, communicating with the other part of the assembly. So again, we have this complete 3D sample. We identify the region of interest, and there is where we go with the high resolution, right, in order to see the cell interaction. And this is important because in these models, we can find, of course, the interactions, but they are not that abundant. So if you take your sample and you start to cut, you have high chances to cut off one of these, and probably you will lose the few connections that you have, right? So like this, what we did and was to validate this type of this pathway connectivity, and the application of this was also published at the end of last year in this collaborative project. Now, something that is also of interest for us and for other groups where we collaborate is 3D cell cultures that are embedded in collagen, right? So if you use the normal digestion methods, protein K is not able to digest collagen, right? So we have to improve this a little bit by combining different digestion steps. So this is a collaboration with the group of Christiana Lungu in Stuttgart, where they have a model of breast cancer cell spheroids. So they seed, right, about 1,000 cells, and then they embed everything in a thick layer of collagen, and looks like this, the sample, right? So if we zoom in, we actually see that the spheroid is that little ball or sphere that is located there, and most of the other part of the sample is just the collagen. So if you do the normal digestion, right, you will not be able to digest the collagen. The gel still will try to expand, and if you make a gel big enough, probably the gel will have enough force to destroy the collagen and expand, but you will be destroying also your cells. You will create deformations, right? So that's why what you have to do is to first do a protein K digestion in order to digest, right, the spheroid, while the collagen is preserving the overall structure. And then you digest the collagen, so then the gel can expand properly. If you do that the other way around and you digest the collagen first, then the gel will already expand a little bit, and your spheroid or your 3D sample will not be able to preserve the original form, and it will be destroyed, right? So it's what we wanted to show is that it's possible to combine different digestion steps, but you always have to keep in mind the order of these ones, right? So here, what we can see is what they are doing. They have like a wild type and that spheroid and a knockout. So the wild type, we can see that is in green. We can see the innervation right into the collagen. The other one doesn't have that. And again, having the ability or having the ability of the 3D imaging is important, because if you see, as you can see in the montage below, right, it's a representation of what will happen if you have your sample and you start to cut it into slices. So the green one in most of the slices will look like just a normal spheroid, and you will not be able to see the innervation that is present, right? Also, when you can have all for example, you can count all the nucleus there. And something interesting is that in the wild type, where we have more the innervation, we have less nucleus than in the knockout one. And we think that the cancer cells, in order to invade the collagen, try to replicate more. But it's still not clear for us like why we have that difference in the proliferation, right? For the knockout cells. The same principle of this collagen digestion could be applied for example, not a spheroid, but just a normal 3D co-culture cells. So here we have like pericytes and endothelial cells, right? So again, normal protocol will look like this. So you have to optimize your protocol in order to be able to digest the collagen and extract then the complete information of your culture cell, right? I think that I'm on time. So to finalize, what I show you is optimized protocol for samples that are 1 square centimeter, more than 3 millimeter in thickness, where you are able to achieve super resolution, like 80 nanometers laterally and 300 in the axial direction. We can preserve fluorescent proteins, works with normal antibodies. And as I mentioned in the beginning, when you combine the expansion and light sheet, basically you have like a method that allows you to image your sample over 5 orders of magnitude. Of course, we are not going to the range of the 10 nanometers, right? It's not our aim, right? But it's a really powerful like method or pipeline to be used. So this is not only applicable for neuroscience, for organoids, as I show you. We have also experience with different type of samples, like jellyfish. We have also, of course, mouse brain cells. We have a project with the human brain to study glioblastoma. So if you have any question regarding like protocols, or if you would like to test something, please get in touch with us. And of course, I would like to thank all the team of Martin Schwartz and all the other collaborators and funding. So thanks a lot.
Okay, thank you for this presentation. Does anyone has question?
Yes.
Yes.
Thank you very much. Very nice talk. I'm wondering why proteinase K does not digest matrix gel, which is a protein somehow.
It's because also you have, well, for example, you don't have also only proteins. I mean, you have also collagen, or you could have fibrin. And at least in our experience, if the concentration of those is too high, then proteinase K is not good enough to digest everything. So you would like to either add something else to get that out. Yeah, so basically like that. Or you can also increase the amount of proteinase K. In our case, I would like to mention now that you mentioned the digestion steps. We don't use SDS. And we usually recommend to not use SDS because then you lose a lot of the fluorescence of fluorescent proteins like GFP, T tomato, and so on that are usually protected by its structure from proteinase K. For example, GFP, right, with this beta-barrel structure is usually not digested. But if you use SDS, usually you start to lose fluorescence.
In the same, if I can, in the same direction, so what are your favorite fluorescent proteins for preservation of fluorescence in your experience?
So sorry,
what are the fluorescent proteins that work best in expansion, that are where the fluorescence is best preserved in your hands?
Well, in our hands, I mean, we have tested a lot, for example, with this technique that is called TEDBO, for example, like M. cerulean, EYFP, TD tomato works really good, but we also have tried other like MRFP or M. cherry also is working quite good, but all the neon green and so on. So we really had problems with, for example, with halo tag when we were working with the culture cells, but then it's also possible to digest, but not using proteinase K. We use something different that I don't remember now, but if you want, I can check. But Helotech is and proteinase K is not working good, but you can change proteinase K for something else.
Thank you.
Yes, I have a question. Good morning. Thank you very much for your talk. I'm wondering if you will be able to measure the expansion factor and the virus distortion problem by imaging the DAPI before expansion, because I am doing expansion of the whole NF node, and now I am doing the imaging with light sheet, but it's for me, it's very difficult to make an image before expansion precisely because I'm using the light sheet and there is a lot of scattering before expansion. So how could you do for that?
What we usually do is like after embedding the sample in the gel, right? Then before the digestion, we put that in the confocal. And with the confocal, we take a volume. And then you should be able to find that volume in the light sheet data. So that's what I showed you the slices. So you can try to find like, I don't know if there is any region that is like characteristics of your sample, right? You can also in the future use fiducial markers, or you can add some beads, for example, outside your sample are embedded in the gel, and then try to find that in the light sheet data. And usually, right, if your expansion is working properly, rigid transformation should be good enough to put the volume of the confocal and the light sheet in place, right? And then you can measure.
Okay. Yes, because is that before I use confocal, but after I use a light sheet, so it's not the same technique and this for that. And do you have some possibility to measure the distortion of your sample or not?
Yes. Well, depending the structure, we can use different methods. I mean, for example, when you work with mouse brain, it's quite easy to find a region, let's say dendrigyrus, right? Like where most of the nucleus are like about 10 microns, and it's really precise. Like the difference is not that much. So you can go there before and after, and then just see how the like sphericity of the nucleus and the size, and then you can get the expansion factor. That will depend more on the type of samples that you're working on. But yeah, I mean, if you want, you can also send me a mail, and we can also talk about that.
Thanks.
I have a question. It's okay to ask? Yeah. So brilliant talk, excellent images. I enjoyed it. How do you stabilize your samples for imaging? I mean, we were trying to do here light sheet microscopy or like expand the specimens with our collaborators to be smoother. We're trying to improvise, but how do you stabilize them? Because your images look spectacular.
So so we have an upright system. So and we just glue the gel in. So the gel is inversed in the sample chamber, and we just kind of glue, right? It's not glue the sample, but we use it with polylysin. Polylysin is really stable. If you will put it on a cover slip, you can even flip your sample. The gel will be there. And then to avoid like the movement of your sample while imaging, then you have to just reduce the acquisition speed. So you usually, if your light sheet is too fast, then you will generate that movement, especially if you are acquiring mosaic view. But beside that, at least for us, there is no other.
And are you doing some drift corrections?
No.
Not really.
No.
Will you be willing to share a protocol on how you do that, or this is something you would like to keep?
No. Sure. I mean, actually, the one that was published is we show also how to do the mounting and everything. But yeah, I can send you that without problem.
Excellent. The second question is, if I'm still allowed, would chaps work equally well for occluded epitopes? Because I mean, it's this Switter ionic, both negative, positive, net neutral, net charge, but less micelles. So I think it will preserve the very exposed epitopes, but the ones that are not exposed, or the epitopes that are found, for instance, within on the luminal part of a crowded small vesicle. Does it work equally good as Triton, or better? Or you didn't look into that?
I haven't tried with vesicles. But I want to clarify that we only use chaps for the permeabilization, because chaps is not good with some antibodies.
I fully understand that, of course. For the permeabilization step, would you still be able to expose occluded epitopes that are buried deep within compartments, or you didn't look into that?
Yeah, that's what I haven't looked into those ones. So yeah, not a good question. I haven't tried that. So, but just wanted to be clear that we did just for the permeabilization, but for antibody staining, we use Triton for the blocking buffer, right?
Thank you.
So before leaving, I know that we are run of time. I would like to confirm with you that the analysis and 3D you do is with Imaris software?
Yeah.
Okay.
Imaris or ImageJ, actually. One advantage of the images that I show you is that most of the images that I show are without any deconvolution or something. Only the ones from where we did the analysis of the pre and post synoptic distances. But the rest of the images that I show are just how they look after getting out of the light sheet.
Yeah. To give an idea to everyone that the Imaris, we are going to have a little practical course of Imaris. So like that, you have an idea what you can do with the software. It was a really impressive work with that.
Thanks.
So we can go to the break, coffee break.
Yes,
please.
if there is no more question, we can have the coffee break. We will restart again the session at 11:30.
Okay. Okay. Can you see my screen? Yeah?
Yeah.
Okay, great. Okay, so good morning, everyone. Yes, so today I will present you. Most of what I'm going to present you is already published. It's more on the application of this cryofixation coupled to expansion microscopy. So I will go through some technical specification and also some application that could be useful for all of you. So it's kind of a catalog of what it's feasible by coupling expansion to cryofixation. So just as a background, I like to introduce this technique with such a way because I'm a cell biologist by training, and I always been fascinated by actually just how the organelles looks inside an animal cell, here a mammalian cell. And I am obviously not the only one, as you can see from this picture, that since decades now, you know that people, scientists, have been super excited on visualizing their favorite organelles, and this is why actually the super resolution microscopy has been developed in general. So here is, I like this review because it really like cover different subcellular compartment of the cell and how they can be observed regarding with different type of modalities. So this super resolution microscopy class is, yeah, okay, so you probably all know that, but it's, the idea is to technically increase the resolution limit beyond the light diffraction to have a nanoscale view of the sample. And we can name many of these techniques, the STORM, STED, SIM, and I think that now it's quite accepted since a couple of years that expansion microscopy can be included into this, this family. Here I'm just re-showing you, but I know that the topic of expansion itself has been very well covered on Friday, but just to keep an element that people have been looking a lot, either by super resolution, the synaptic contact, and by expansion. And I would like to compare this light super resolution microscopy with the electron microscopy using electron, as his name tells. And here you see exactly the same compartment, this synaptic contact with the pre-synapse, where you see the single vesicles, and the post-synapse here adjacent to it. And this technique of electron microscopy is actually coupled to cryofixation, that I will come a little bit later on this. And this is because of this specific type of fixation that the resolution of the electron microscopy has been further improved. As prior, when using chemical fixation, it was disturbing and some of the, notably the membranes, and that's why cryo-electron microscopy has been developed also a couple of years ago. So the electron microscopy is using a different source of light, of energy to illuminate the sample, here the electron. And if we list the pros and the cons of both of these techniques, here you can see that the light super resolution microscopy has, of course, the power of being resolutive. It allows for nanoscopic localization, but very often it's constrained by a chemical fixation that is necessary, although we can do live super resolution microscopy, but if we stick to fixed sample, we need to add a chemical. And of course, it's protein specific in a way that you have to stain for 1, 2, 3, or a little more now, but you're limited in the sample that you want to look at. On the other way, we have the cryo-electron microscopy that is, of course, resolutive. And but on top of that, it has the advantage of being native because of this cryofixation method, not using chemicals. It allows to have a cellular context, as you could see from the previous image. But usually the labeling are extremely complicated in cryo-electron microscopy, so you cannot really look at specific proteins, and it requires to work in cryogenic condition. So when I started this project during my postdoc in the laboratory of Paul Guichard and Virginie Hamel, we wanted to gather all the pro and get rid of the cons. And this is how actually we thought of cryo expansion microscopy, because with this technique, and I hope that I can show you this, that we only combine the pros, like it's resolutive, allowing nanoscopic localization within the cellular context in native condition. And I think the power of developing this technique was also in the way that, like, if you really look at all of these images that are acquired with different super resolution modalities, now with expansion, we could see that it's feasible to see all of this same organelle or compartment with only one technique. With here some just slight modification. This image of the nuclear pore and the top view of the centriole have been acquired using iterative expansion microscopy. But it's just to show you that how simple it is to look at anything you want to look at. And I think that the other talk of this morning also proved this anyway. Okay, so the question that we're asking when we developed this was like, okay, is it possible to avoid chemical fixation artifact? Can we perform multicolor and high-resolution imaging? Catch dynamic event, I really like, don't put too much emphasis on this because, of course, we are doing fixed microscopy, but you will see that it's pretty nice when we combine with cryofixation how the dynamic element we can catch. And also, by the use of this light microscopy combined with the cryo, we can have a cellular context in which we precisely localize our proteins. Okay, so I'm not gonna go back to the principle of expansion microscopy. I think you heard a lot and you will also practice this next week. So I'll go super fast on this. And of course, if you have questions, we can always come back. The only thing I want to insist is that the protocol that I am going to present is the post expansion, or ultrastructure expansion microscopy, meaning that we do the immunostaining after we start the process of expansion. And I just wanted to highlight some little tricks that that is really like a protocol thing that we actually are gathering onto a Jove manuscript that I hope you'll be able to get access very soon. It's just to show you that at the moment of the denaturation, when I joined here the lab in Lyon, they were already doing expansion on Drosophila, and they used this little biopsy punch, extremely convenient, to delimitate the area of the gel that you want to expand. Of course, that you can expand the whole cover slip, and this is what you can see here. That's the whole gel from a 12 millimeter cover slips. But it's quite convenient also to punch and only take these little pieces that you can directly proceed for a denaturation, and then the stainings that is done into a plate with silicone. It's super convenient also to reduce the amount of antibody. Although you can also cut your gel in little quarters, and it's also a way to reduce. But I really like this strategy personally because it costs less. Of course, the expansion microscopy is extremely costly in terms of antibodies, so it's always good to limit. Okay, that was a little parenthesis. Now what I want to focus on is this cryofixation step. So I mentioned that it was a method of fixation that was developed in priority for electron microscopy study. So we just completely took this way of fixing from this field. And the idea is to plunge your sample with high speed in liquid ethane at minus 180 degrees that will produce a vitreous ice without any crystals. And this is the best method to preserve the ultrastructure of the cell in a native condition. To do that, when I started the project in 2000, on this specific setup, and you just arm the arm. You can just activate it by this little button, and then it will plunge into containment in which I just put liquid ethane, chill by liquid nitrogen. Here I just want to show you how it was when I started at the slow motion of the setup being activated. So as you can see that the organization of the whole setup was completely empirical and not extremely well established. You could see that the movement and everything is like jumping because of the action. And so since then, we have now a company that developed an adapted manual plunger, although you could use any manual plunger from an EM facility, but it's also quite convenient to have one that is adapted to our sample. And in this one, everything is perfectly aligned, and we have this little element here that allows to demonstrate the level of liquid nitrogen. So this will just rise up, floating on top of the liquid nitrogen, and then you just plunge here in the middle of this element here. So it's pretty convenient to use this one, although I really like that I developed the project using the Jacques Dubochet, the Nobel Prize for cryo-EM. We get lucky enough to have this in our lab. Okay, And then the step that what we don't want, of course, is to stay in cryogenic condition. So we need to go for a step, what is called free substitution. And for that, we just dip the sample into extra dry acetone that will replace the water molecules inside the sample as soon as it thaw. So basically, what we do after we cryofix the cells, we just transfer them onto these little tubes here that contains acetone, which is completely frozen. We just put everything on liquid nitrogen. So the acetone is frozen at minus 180 degrees. So then you just lay down the cover slip on top, and then you incubate this on dry ice overnight. Although this can be reduced, but it's quite convenient to do it that way. And once the temperature reaches minus 100, more or less, the acetone melts. And then as soon as you continue to rising up the temperature, as soon as the ice is becoming water, it's replaced by the acetone that has a diffusion coefficient higher than the water. Then once we are going back to room temp, we proceed to rehydration just by replacing the acetone first by 100% ethanol and then by a bath of increasing water concentration until a water-based solution that is compatible with expansion. And then we just go for the classical protocol with the acrylamide formaldehyde anchors and blah, blah, blah. Okay, so the first thing that we wanted to do, of course, was to show, okay, so we could do the cryo, but how was it compared to classical chemical fixation? So with this, we always compare the cryo to 3 other ones, either the paraformaldehydes, methanol, or the mixture of paraformaldehydes, glutaraldehydes, which is the gold standard EM way of fixing because it's extremely strong and preservative. And for that, we compare the tubulin. So you can see that the microtubules are not well preserved in all the conditions. It's extremely poor in PFA after expansion, while it's quite okay on methanol and nice with glutaraldehyde. And then to also test the universality of this protocol, we combine to something very different, the endoplasmic reticulum. Again, in these 3 conditions, and as you can see here, methanol and PFA are really poor preserving the membranes, while the PFAJ is quite okay as published. But if you zoom in a little bit, we realize that the glutaraldehydes didn't seem to be enough after expansion. We could reveal some fracture of the endoplasmic reticulum. And then we applied the cryofixation. That was absolutely gorgeous for the microtubule and also relatively nice for the endoplasmic reticulum. Then all of this image is the convolve image from Whitefield, the convolve image. But with this deconvolution, we are losing the intensity element within this sample. And when we compare the cryo versus glutaraldehyde PFA, regarding the intensity, we could see that the intensity was improved in cryo, which is a known drawback of the glutaraldehyde, that it quenches the fluorescence. So we had something similar in terms of architecture of the ER, but also in terms of intensity. So this is two advantages of using the cryofixation. Then when I mentioned that we could catch dynamic events somehow, I meant that we could look at the relationship or the communication between the ER and the microtubule. And we know that that has been published using LiveStead, that the contact site between these 2 organelles is extremely dynamic. Like this touching point at the tip of the microtubule doesn't last more than seconds. So we're quite happy to see this touching behavior or even the sliding. I mean, of course, it does not slide, but it looks like it. And that was really for us a proof that we are really like snap freezing these cells as the moment that is doing this. Then we wanted also to see other type of organelles. So we looked at mitochondria that were generally well preserved using glutaraldehyde without any surprise. And really poorly preserved in methanol. This is known for anyone working on mitochondria. Although our pre-FA fixation here is really bad, I heard since we published the paper that you could have little tricks to better preserve the mitochondria with PFA. So I think that, I mean, this is really like the classical way of doing it in the lab, but of course, I completely agree on the fact that this could be improved using chemical fixation. So with this, we just looked at the mitochondrial area, and we could see that cryofixation was equal to glutaraldehyde regarding this parameter. But I think that you can appreciate here that the intensity seems a little different. We have much more background here because I had to push a lot the signal using, I didn't say, but it's a mitochondrial matrix marker. And also we compare the antigen accessibility just by counting the number of TOM20 dots. So it's an outer membrane proteins. And we could see that the cryofixation was really going above the PFAGA fixation. So again, this cryofixation seems to combine two advantages like that or to alleviate some drawbacks of the glutaraldehyde regarding also the masking antigenicity. And then just to show you a little more example of the mitochondria staining, we're also quite happy that we could see the cristae of the mitochondria. Here again, using MitoTracker, localizing inside the matrix. And just to give you a little bit of resolution we can reach, we can really decipher between the different cristae. So this could also be achieved with chemical fixation, but we also had to show that, yes, it was better in some way and in other way, at least equal. Then something we really focused on for a couple of months was the expansion of mitotic cell, because it was known to be quite complicated to first having a full mitotic spindle expansion because it's extremely rigid, especially, and also because you have to chemical fix to preserve, for instance, here the astral microtubules. And we are super happy to see that we could actually nicely preserving them without using a super strong chemical fixation that would prevent the expansion. And here we just quantified the fact that we could have a 4x expansion, while in PFAGA it was known to be limited to like 2.5, 2.6. Another example here of a mitotic cell, and I just chose this one because I wanted to highlight on the fact that there are some artifact or stuff you need to be careful of when applying cryofixation, and this is not only the appearance of this crack. This is quite classical, even EM people described it quite a lot. And here I just wanted to show you another example of a bigger crack here that if you look on this zoom in area, it doesn't really disturb the overall architecture of the organelle. So having cracks is not a sign of bad fixation. It's just that it sometimes happens that you have one crystal and that it disturbs the sample, but the way the cells look around are usually not really damaged. However, there is one strong indication that cryofixation was like badly performed, and it's the appearance of this bubble here that you see inside the nucleus. This really is a sign that the temperature of the cryofixation was not properly controlled, or that the sometimes can happen that the sample was not perfectly immersed into the ethane. And this is really, really like catastrophic for the cell. So if you have this type of, sorry, if you have this type of bubbles, you just have to discount this sample and work on the fixation condition. Okay, that was just a little parenthesis because I realized I was presenting a lot this technique without mentioning any of the, things that you don't want to see. Then I want to go back to this cell because we can super nicely appreciate the centriole, and you probably know that Paul and Virginie's lab is completely focused on the centriole. So of course we could not looking at them to see how they look by cryo, and they look quite okay. So here you have a not a side view of one centriole, and here the other one is completely orthogonal. And we used this also to show that the expansion of this organelle was not altered by the fixation. So I could show that both the length and the diameter of this centriole, when compared to cryo-EM data of this organelle, was like completely fitting with a 4x expansion. Okay, okay, so the one question that we kind of always ask, okay, can you see dynamic events? I show some regarding the connection between microtubule and ER. But actually, I wanted to show something that was really important for us. It was regarding an artifact that was known in literature regarding some transcription factor. Here we have SOX2, that when you look at these cells in fixed condition, in mitosis, it seems that the SOX2 stainings is completely getting away from the chromosomes. And that was believed that it was true for many years until people start doing live. And when they realized that doing live was showing a completely different pattern of the localization of SOX2 that is remaining on the mitotic, on the chromosomes during mitosis, they showed that actually it was probably an artifact from the PFA that was retaining the transcription factor within the cytoplasm. So we wanted to verify whether cryofixation could reproduce the live and therefore avoid this artifact. So we did the SOX2 staining in non-expanded conditions. So it's also a good point to show that you can use cryofixation without expansion as well, although you're all here to do expansion. So yeah, I guess that if one day you try, you will combine both methods. But it's nice to see that, okay, we reproduced the PFA fixation, getting rid of the SOX2 into the DNA, while we could keep it using cryofixation. So cryofixation is really like an equivalent of live conditions somehow. Then I told you that, okay, one of the disadvantages of light microscopy is that you cannot have a cellular context. So this, you probably saw images on Friday on the iterative expansion microscopy showing a lot of this cellular context labeling, the NHS ester. And here I used it to mark to label the mitochondria, and here you have just the DAPI. And by combining this with normal stainings, it brings a lot of information. Here you see two images, one image of two cells harboring a primary cilium. And I will come to this organelle because this is my new favorite things to study now that I am in Lyon. And here you have a just a tubulin staining. You see here one cilium and here another one. It's quite difficult to see, where they are, where are the cells. And by combining this tubulin staining with NHS ester, that is becoming quite clear where these organelles are localized. And actually, what is quite nice with NHS ester, that it really reveals a lot of stuff, like you nicely see the nuclei, the mitochondria, the Golgi apparatus, and here it's even endolysosomes. And then, so if we zoom on this cilium, it's quite nice to see that actually the cilium is always localized in the vicinity of the Golgi. And without this combination, it would have probably been difficult to co-localize these two together. So that's why also we really like this method since a year. With this, I'll reaching the end of the talk. I just want to show you all the application of the cryo expansion microscopy, some of which you already heard. One parasite, the Trypanosoma brucei, that is actually a collaboration with a lab from Bordeaux, the laboratory of Mélanie Boniver, that I did with Héloïse Berthiaud on the Trypanosoma. So we just proved that we could cryofix and expand it, and it was extremely gorgeous to look at. And now Héloïse is actually developing her hand project on this. Then, as Jim told you on Friday, we could also expand the yeast. So I'm not going to go too much on this because he also showed these images. Regarding the plant, the only thing that I could try with green algae, Chlamydomonas renarditi, because it's a very canonical model for ciliary studies. And again, the cryofixation is working tremendously. Here you see the thylakoid stacks, and the mitochondria is really nice, and it was published 2 years ago. And also something a little bit more experimental is we could test the cryofixation on environmental sample. First, we did a pilot experiment in Iceland with people from EPFL and the MBL, and the idea was to fish sample from the Iceland Sea and to try to expand cryofix and expand them. And actually, this is now being gathered together to show you how nice it can be. All of this I could not put in this recent publication. I just wanted to list it that are now using this cryo expansion in the parasite field on Toxoplasma that really changed the way we see this little parasite. Other paper from the green algae Chlamydomonas. So this one I mentioned it, but there is this one on the organization of the cilium. Other papers on yeast, of course, not now. I guess that people will continue to use this because it's pretty convenient. And two paper on close relatives animals and environmental sample. Something I didn't mention, but regarding the yeast and these close relative animals, the use of the manual plunger is rather limited because the sample are a little bit too big or they have the shell that is sometimes difficult to cryofix. So with this specific sample, it's better to use a high pressure freezing machine that you need an EM facility, of course. So I didn't put a lot of information about this because I wanted to present you something that you could do directly in your lab if you don't have an EM facility nearby. And finally, just two very recent example of use of this method in mammalian cells to look at HIV infection, and recently on the organization of immune cells. With this, I think I'm gonna finish. I maybe be a little bit over time. So yeah, I just show that you can preserve the cell structure in its native state, allowing for visual nanoscale localization, that it's quite versatile. It's still low cost and easy implementation because it's expansion, and you can even couple it to super resolution. With this, I am finishing with acknowledging the people. So people from the University of Geneva, my former lab, and the people I'm working with now in Lyon, and some collaborators. And I am actually developing a super cool project on cryofixation expansion of zebrafish with Julien Vermeau in London. And yeah, I'll thank you for your attention, and I'll be happy to take your questions.
Yeah, thank you very much for your very nice talk. We have just time for 2 questions, I guess.
The participants, if it's possible.
Mm-hmm.
I could have a little question waiting for their questions, because we use a lot of also cryofixation. What do you think happens when you cryofixate? Why, even if you put then acetone, ethanol, and then, or now that you modify protocol with glutaraldehyde and formaldehyde, why do you preserve better the structures when you compare to just chemical fixation? What is happening even for the cryofixation? Because you are going to use things and they are very similar to chemical fixation. At the end, you are going to fixate chemically.
I think the key, and here I'm just going to quote old papers that I was reading again and again because it looks like magic. But to me, the key is the fact that the free substitution is happening at low temperature. So I think that's the key, because then what's happened is that you have this vitreous state that really preserve the elements as this is something you don't have with chemical fixation, because they are slow fixation. I think 2 points: the rapidity versus the slow fixation. That glutaraldehyde takes 20 minutes, super slow, so then you have time for the sample to start dying a little bit. And also the fact that we do this free substitution at low temperature, it really limits the Brownian movement of the sample. So what's happened really, like as soon as the ice is starting to melt into water, it's directly replaced. And I think the fact that you have this very slow movement within the sample, it also prevents the disturbance that you would have with chemical fixation. This is what I gathered from Kallenberger paper, like from the 70s, but he really described it like this for the origin of the cryo-EM. So this is how I understand it.
Mhm.
I have a question. I didn't quite understand the slide with expansion, the cracks. How do you know that it doesn't affect cells and structures?
Yeah.
Because you show the crack and the bubbles, but for me, when there is a distortion like that, a crack, it affects the structure.
So yes, at the exact position of the crack, of course, it's impossible to analyze. But what I wanted to say, it's the first time I was really presenting these bubbles and crack stuff, so like, maybe I was not super clear. What I mean is that the cracks is happening before the expansion. It's not an expansion artifact, it's a cryofixation artifact. So you really have the cell that's split in two. So of course, at this position, you will have the loss of your information. But what I was always kind of amazed is that on the dividing cell, the mitotic one, we could see that the rest of the cell was completely preserved. Like as soon as the crack has been done, then the rest of the cell is nicely cross-linked to your gel, and you have a normal expansion. So to me, and I was super scared at this at the beginning, but then talking with EM people, they said, yeah, it's completely normal. It doesn't prevent today. But then the bubble is completely different. It's really like a super bad either a fixation or free substitution.
Okay, it's a consequence of the cryo-EM. Okay, thanks.
But there is a question in the chat from a participant. They ask whether the ROST device is already commercialized.
Yeah, absolutely. Yes, I can on this because if I send the PowerPoint, I can add a reference on top of it that you can have it if you want. It's commercial. Yeah, it's available and it's around 5,000. Just saying that you can anticipate if you want it.
So you're gonna see in the demo that someone here is gonna do in next week. The problem is not the device actually, because we are gonna use an old device, like you were saying, this Duverger device. Is mostly the ethane and the freezing of the ethane and the disponibility of this gas in the lab. It's gonna be mostly the issue that you're gonna have to find around the possibility of having this because well, the little device for the Vitrobot, we use it also, but it's also the ethane freezing, because you have to. That is gonna be the only thing, because it's also hygiene sécurité. It's something that's gonna be a little bit complicated to implement. But Eloise did already here in Bordeaux, so she's ready to do. She was doing the cryofixation here, and now she's autonomous and she installed that. But you need a little time to.
Yeah, it's true that the gas. I didn't mention that, but yeah, the gas. If you're absolutely not an EM lab, it could be quite difficult.
In France.
And at some point you manage, but yes, getting organization, the safe place to install the system, also you have a lot of ventilation and stuff. That's something that needs to be considered. But
I have a question if we still have some time. I don't know.
Yes, maybe just quickly.
Yeah, just regarding the bubbling that you say it's a bad cryofixation. Do you know, or can you advise on some settings that we can handle to improve the cryofixation method?
Yes, I think that my experience is that the bubbling is coming from the the fixation step, really the moment the sample enters into the liquid ethane until the moment it's on the acetone. What can cause this is the remaining medium or whatever solution your cell are in, that you really need to blot seriously and rapidly, of course. The temperature of the ethane itself. So you either can combine the one from Rost is without temperature control, but you could also have with on temperature control. With this, you're completely sure. But what is super important is to wait at least, I would say, 15 minutes for everything to be like really like at good temperature. And the last is the transfer between the liquid ethane to the acetone. This you really need to be super fast. If you can stay, it's true that I could make a video about this. Probably soon will be online, I hope. That you can, the moment you transfer is super, super important. You have to stay in this little gas state where it's still cold and you just drop as fast as possible your cover slip. To me, that's the 3 moments something can go wrong.